Hi, everyone. Great to be here. I'm very happy to talk to you, and very nice to meet all of you virtually. I'm Nikolay Oskolkov, a bioinformatician from Lund University in Sweden. And today I'm going to talk about our recently published computational method, which is called aMeta, which is an ancient metagenomic profiling workflow. And it can be installed from this GitHub page. And this is a great team behind aMeta. So I immediately express my gratitude to all my collaborators. And aMeta is about ancient microbiome analysis. So we are going to talk now about ancient microbes that can be detected from their DNA traces present in human or non-human remains coming from the past. So, for example, if you are working with a mammoth data, if you have a mammoth task, what people typically do, they drill and extract DNA from the powder, and then you can sequence this DNA. And what we analysts usually get is this mixture of DNA fragments, or what we say reads. These reads come from different organisms. Of course, since you've been drilling a mammoth task, you would expect that the majority of reads will be coming from the mammoth. But then accidentally, we modern humans can drop our DNA. So some of the reads might be coming from human. And more interestingly, some of the reads, or I would say substantial amount of reads, usually comes from microbes. And then if we can show that those are ancient microbes, so microbes that coexisted with the host, it can be interesting because it can tell us a lot about the lifestyle of the host in the past. And there are different ways to demonstrate that those are actually ancient DNA fragments, but maybe the most convincing and straightforward is so-called damage pattern or gemination patterns. It turns out that there is an enrichment of C2T and G2A polymorphisms at the ends of the DNA fragments due to some weird chemistry going on. So this damage pattern is a good evidence to prove actually that this is an ancient microbe. So this is how they detect ancient microbes in archaeological samples. And some of these ancient microbes can be really interesting. They can be pathogenic, for example. So already in 1998, there were studies reporting the presence of Yersinia pestis DNA structures in dental pulp in human samples. And Yersinia pestis is a plague-causing microbial agent. And since then, when the next generation sequencing came, so there were many studies detecting different viruses and bacteria, interesting pathogenic viruses and bacteria in different archaeological sites and samples. So it became a very popular area of research, and many labs routinely screen their ancient samples for microbial pathogens. But however, very quickly, people realized that this area is full of false positives, that it's very sometimes you might run into troubles if you're doing a naive analysis. For example, what we are showing in our paper, and other people also show in different studies, is that if you take just a random metagenomic sample and map the sequenced reads to Yersinia pestis reference genome, what I'm doing here on the y-axis, I'm demonstrating the number of reads from a modern infant stool metagenomic sample, random sample, that map uniquely to Yersinia pestis reference, even though they were given a choice to be mapped to human reference genome and many other microbial reference genomes. So despite that, they still prefer to be accidentally mapped to Yersinia pestis, and it can be thousands and tens of thousands of reads. So that just the number of reads mapped to Yersinia pestis reference genome is probably not a good evidence for its presence, because it's unlikely that modern infant random infant had plague. So that's a negative example how that demonstrates how easy you can run into troubles here if you're doing a naive analysis. So in fact, if you would have looked inside the alignments, you would have detected, you would have realized that the reads map in this so-called non-uniform or uneven coverage way. So typically what people say in this case is that these reads, they actually originate from other than Yersinia pestis microbes, but they map to conserved regions of Yersinia pestis, although they are from other microbes. So that's an example how people see and detect that it was a false positive discovery. And this was exactly the goal of aMeta, is to minimize the amount of these unfortunate false positive discoveries. And we achieve this goal following three key principles, such as we are using very unbiased competitive mapping, and we're a little bit extreme in this sense. For this purpose, we need very large and diverse reference databases. And we are also specifically controlling for evenness of coverage. It's our major criterion. And I'm going to quickly go through these key principles and explain what they are and why we think they're important. So why do you need competitive mapping? Well, if you do, maybe if you have a background in genomics or doing human genetics or population genetics analysis, maybe this competitive mapping thing seems unnecessary for you. But so, for example, if you work with like from the previous example with mammoth data, and you have sequencing data from a mammoth tusk, you can align those reads to elephant reference genome as a proxy for mammoth reference genome. But then if you're a good analyst, you would suspect that some of these reads are not blue, but actually green. So they actually come from maybe human. You would expect some percentage of modern human DNA to be present. So you at least need to do a good analysis to actually disentangle human and mammoth reads. You at least need to concatenate their elephant reference genome with human reference genome to actually disentangle those human and mammoth reads. So if you think even deeper, you would expect that actually there can be many microbial reads present there. So in order to do even better analysis, you would like to actually concatenate the elephant reference genome with human reference genomes and with many microbial reference genomes in order to do more accurate analysis. And this is what is called competitive mapping, which is absolutely central for not only ancient metagenomics, but also modern metagenomics field. And I hope you can see what I was doing here. I was actually increasing the database size by constructing this grand reference genome, concatenating the elephant, human, and microbial reference genomes. I was increasing what people say databaise size. And that's exactly, that's very true that in order to do a better analysis, you need big databases. So that's a second key principle of aMeta. So what we show in our paper is that, based on simulations, is that bigger databases, they provide a higher analysis accuracy. So on the y-axis here, I plot the Jacquard similarity intersection over union between the simulated microbiome and the reconstructed microbiome. And it turns out that in order to get a better reconstruction of simulated ground truth microbiome, you need to utilize larger databases. So the third point was about evenness of coverage. So why it's important? I already touched upon it when I explained the example with false positive Yersinia pestis in a modern infant sample. But just to reiterate, so here are two mapping scenario. If you just compute the coverage, what people typically use as a definition of coverage, those two scenarios, they have an identical coverage or depth of coverage. So using this as a criterion, on the number of mapped reads as a criterion, you would say you detected a microbe in both cases. But it's also important to see how, not how many reads map, but also how they map to the reference. And in the first case, we say the microbe was not detected, while in the second case, we say microbe was detected. We actually want the reads to evenly cover the whole reference genome. So that's important. And that was about aMeta, but also if you follow this field, if you're into ancient metagenomics research field, you probably can say, so what about other tools? Maybe there are other tools. And yes, there are other tools. I wouldn't say there are many tools, but there is maybe one or two other tools in this research area. And for example, HOPES, which was published in 2019, a very good tool. It has a very nice design. It answers two questions: who is there and how ancient. That's exactly pretty much what you want to know in ancient metagenomic analysis. Unfortunately, it has severe limitations. For example, it's very resource demanding. So we are talking about literally about a few terabytes of RAM, which should be used as a routine, and not many labs have a few terabytes of RAM on their computer clusters. Also, so this leads to this constraint that HOPES and its internal aligner, which is called MALT, are only feasible to use with limited size database. And as I showed previously, small databases are less accurate. So you sacrifice accuracy if you use limited size databases. And also HOPES completely ignores the evenness of coverage. So that's all. This is all we wanted to fix in aMeta. So we wanted to overcome these HOPES limitations. And this is a brief overview of aMeta. I will not be explaining in details how it works, but just to emphasize that the trick behind aMeta, why we actually use less resources, and in this way we can use larger databases in aMeta compared to HOPES, even though we still use MALT. But we pre-screen what we call pre-screen our raw data with Kraken Unique, which is the recent development can use as large database as you want, and it can be run literally on your laptop. So we in the first step we are very unbiased. We are utilizing huge databases, and we pre-select only reliable candidates which are likely present in your samples, and we dynamically build a MALT database for each project and propagate those reliable candidates to the MALT, which so we still need proper alignments in order to do abundance quantification, microbial abundance quantification, and also validation and authentication analysis. This is how the output of aMeta looks like. So I'm not going to explain this all these plots. It just to emphasize is that we are not using just one criterion, one metric as a criterion for ancient microbial detection. So we actually use a combination of different metrics in order to reliably conclude that a certain microbe is really present and is ancient. So all these metrics are on the left-hand side, and on the right-hand side is a heat map, a summary, what you typically get from aMeta. So we try to quantify these plots on the left-hand side. We try to quantify them and digest them as scores. So we create a scoring system so that the users, they can get this nice summary, this heat map where they immediately can see what microbes are present and ancient in what sample. And also, since we suggest a new method, aMeta, so we wanted to do a formal comparison with HOPES. So this was done extensively using different simulated datasets, and also real data. So here, what I'm showing is that we simulated microbial sequences, ancient microbial sequences, and tried, so this is a ground truth that we simulated. We knew how much of each microbe was put in each sample, and we tried to reconstruct this microbiome, and we compared aMeta versus HOPES in terms of detection accuracy and authentication error. Detection and authentication accuracy. So detection error, so those are two major contributions in analysis error. The detection error is when you misidentify microbes regardless of their ancient status, and authentication error is when you detect a microbe and assess it to be ancient while it's a modern contaminant, for example. It's an authentication error. So in terms of both detection and authentication error, what we show in our paper is that aMeta outperforms HOPES. And also importantly, we compared resource usage of HOPES and aMeta, and we show that aMeta uses at least twice less memory compared to HOPES, which is important for utilizing bigger database and get a better accuracy of our analysis. So that's pretty much all I wanted to show you. So I would like to thank our team, which is a National Bioinformatics Infrastructure Sweden that I belong to, and I thank you for your attention.